Yuval speak. Professor Yuval is an investigator at Aviv University and the director of the Gold Brain Institute at Ichilov. He did his PhD in the Weizmann Institute with Rafi Malach and a postdoc at the Center for Sleep and Consciousness with Judy Tononi and Chiara Cirelli at the University of Wisconsin-Madison. He opened his lab in 2012, and research in the lab focuses on sleep and sleep-like states, relation to cognition, and so many things to cover. We can't even start to cover all the amazing things he does. So we're looking forward to your talk. Thank you for coming.
Toda rabah. No microphone or anything needed, right? You can all hear me? So first of all, thanks everyone for coming in such large numbers. It's very nice to see a packed room with interest, and it's really an honor and pleasure for me to visit this place. You may be used to it on a daily basis, but when entering this beautiful building, this court, it feels almost like a temple for brain knowledge, and it's in a nice way. So I'd like to start with two apologies. One is that in my talk today, I won't be discussing new fundamental insights on how the brain works. In the last few years, we've been invested in developing some new tools, and I think these tools are very exciting, and I think they have the potential to drive new discoveries. So some of the talk focuses on these new methods, these new tools, which I think are still quite interesting. And the second apology is that I put too much into the lecture and into the abstract. And actually a good friend who I met this morning advised me, Yuval, you're crazy. This is like a job talk where you're trying to prove that you did this and that and this and that. You shouldn't go this way. So very good advice. And I decided last minute to cut some of the content that's in the abstract, the part about more rodent, locus ceruleus, noradrenaline activity. So for all my colleagues here, Mickey London and others, I'm sorry for the disappointment. Okay, so after we got this out of the way, we can begin. So in our lab, we study sleep in both mice and men. And we study a few different themes. One is what we call sensory disconnection. In what ways does the sleeping brain respond differently to sensory input than when we are awake? The other one is how sleep supports memory consolidation. We're also very interested in arousal, and specifically the system in the brain that secretes noradrenaline through the locus ceruleus and other nodes. And also sleep as a powerful window into brain health. And today I'm going to focus on I'm not going to talk at all about sensory disconnection or auditory responses. I'm going to talk about the other domains. So in the field of sleep and memory, in humans, in cognitive research, usually the paradigms are such that people come in the lab. Before sleep, they learn some new material. Usually it's word lists. So let's say projector and pine tree, or wooden desk and a red marker. And then they go to sleep, and then in the morning we test their ability to recall their associations. This is very standard, but also, first of all, it's very boring for the subjects. Now we work more with elderly patients, with people with cognitive issues, and it's really quite a challenge for them to do these tasks. Also, it's very much relying on language, and it's very different than how we study memory in lab animals, such as rodents, where we infer about memory indirectly from the way they explore their environments. So we wanted a method that sort of brings these worlds together and is more natural, is more engaging. It doesn't rely so much on language and report. And we thought this work is led by Flavio Schmidig, a wonderful postdoc in the lab, Daniel Yamin, and Omer Sharon. And the idea was that could we actually come up with what you can call a no-report paradigm for memory, for even episodic memory. So for, let's say, in consciousness research, Leon knows this very well, people report about whether they consciously perceive some stimuli, but this introduces a lot of confounds of reports and decision-making, and there's a lot of advantage in being able to study these phenomena without report. So we thought, could we study episodic memory without report? It's almost like an oxymoron, right? In all the textbooks and what we teach, episodic memory is part of declarative memory. By definition, it's something we always report. We study it via verbal report. Must it be that way? And the idea we had is perhaps we can use eye tracking and the way people explore visual content with their gaze to learn about memory. And this has been done before we began, of course, especially in familiarity. For example, if you're presented with an image that you've seen before, you're going to scan it with your gaze in a different way than if you haven't seen that before. This is known for familiarity. And our question was, could we drive this further to really study episodic memory, so to study memory of events that happened at a specific time and place, not necessarily autobiographical events, but events out there. And to this end, we took a lot of effort into producing special animations with an animation studio. Let me show you an example. Each animation is about 5 to 10 seconds, and we have 100 of these. And in each, this is a typical example. This is one of the animations. So you see something like this. Okay, and then a salient, surprising event appears, right? It's very difficult to see it coming, literally, in the first time you see the movie. But once you've seen it, and you have a memory for it, you're probably going to look at this movie differently. And indeed, when we, for example, if we record eye movements, this is one particular subject looking at this movie for the first time, they're not going to spend much time before the event in this region of interest that we now denote with this rectangle, right? This is very natural, but once the meerkat appears, we're going to look at it. Very natural. The second time people watch, the same person watches these movies a few hours later. This is now the green dot. And you can see, it's very intuitive. Starts visiting, is he going to jump now? Is he going to jump now? The person is starting to check out that location in the seconds that lead to an event, right? It's very, very intuitive. It turns out it's a very strong effect, and we found a number of ways to quantify this. But basically, it's very simple. We just measure the distance at each time point between where the fixation is and this area or a coordinate where we know the event will appear. And we've prepared these animations in a way that the events appear at different times and at different locations across the screens, and it's all controlled. And of course, the average distance to this event is much lower in the second time we watch the movies than the first time. It happens for each and every person. And what's really exciting is we did a number of experiments where we crossed this with reports. So for example, we had movies that did not have an event at all, or where we stopped it before the event appeared, and we stopped. We asked the people, okay, do you remember, have you seen this movie before? Yes or no? How confident are you? Tell us about the event. Was it this event? Was it that event? point on the screen where do you think the event will happen? And all sorts of questions. And we've shown that this gaze effect is highly correlated with the specific report about the memory. So the more we see this gaze effect, we know how well, or we can quantify the memory, or the memory engram, if you will. And we can also do this without report. We have some experiments with report and some without. It's such a strong effect that, in fact, we can use machine learning algorithms to train machines to take 3 or 4 seconds of data, single trial. So one person looking at one movie, if you give the machine 3 seconds of data before the event appears, the machine can say with 70% accuracy if the person has seen that before, if it's the first time they watch the movie or not, if there is a memory or not. And the reason why we're so excited is now we can, for the first time, perform memory experiments without report. This is just one example out of a few different applications. We can have people watch the movies twice, let's say 4 hours apart. Either they were given an opportunity to nap or not. If they slept, then their memory as reflected in their gaze. We don't talk with the subject, we don't ask them anything. By the way they look at the movies, we can see that if they slept, their memory as reflected in the gaze is at a higher level than if they did not sleep. Right? So this, first of all, it has a fundamental scientific meaning. Because until now, we didn't know, for example, does sleep help memory, or does it help the ability to report about the memory? It's not the same thing, right? So now, when we have it without report, we can be more confident that it's really a memory consolidation that we're measuring. And the nice thing is, for the first, I'm not sure for the first time, but we have now a method that we believe has a lot of potential to study memory in all different scenarios without report. In babies, in non-human primates, right, Didi? Maybe the monkeys can look at these movies and we can measure hippocampal neurons with the same movies that we have established that the people report that they remembered. Maybe we can use that with people with aphasia. Or after stroke. Let's say someone has a stroke and now they're at their bedside at the hospital. We don't know if it influenced their memory, because we don't have tests to measure memory, hippocampal dependent memory, without report. And I can give you a teaser that we're now continuing this to see to what extent this type of memory is hippocampal dependent and reliant on the medial temporal lobe structures. And we have some preliminary evidence showing, both with fMRI and with intracranial measurements, we are even bringing brain damaged patients like the Israeli HMs to watch these movies, and this is sort of where we're going. What are the brain mechanisms that support this gaze memory? Okay. Another gap between how we study sleep and memory in humans and in animal research is, of course, our access to the relevant brain regions, right? In human research, the hippocampal activity is poorly accessible, and the human studies rarely allow for causal interventions that perturb the mechanisms. So also we wanted to push the limits of what can be done in this domain in a very unique project led by Maya Geva-Sagiv when she was a postdoc at the lab in collaboration with Yitzhak Fried. We work with epilepsy patients. We've been doing it for some years now. These are drug-resistant epilepsy patients who do not respond to the available drug treatments. They're often implanted with depth electrodes to monitor their seizure onset zone for potential treatment, and many of them volunteer to participate in experiments. This was the case here. And here we wanted to specifically see what happens in the dialogue, so to speak, between the hippocampus and the neocortex during sleep that supports memory consolidation. There's a lot of theories based on animal models that posit that this synchrony or communication, especially around ripples and replay events, is very important for memory consolidation. We wanted to test this in humans. Could we causally enhance this synchronization via deep brain stimulation? Could we evaluate how this affects the memory? And also, what does this do to the neuronal activity at single neuron resolution? So this is the setup. We have on the right these depth electrodes that are implanted. This is a post-implantation CT of a renal patient. And you can see in the middle are all the, for an aggregate of many patients, these are the regions in the medial surface of the brain that we typically monitor. It's especially the medial temporal lobe, hippocampus, entorhinal cortex, amygdala, but also along the cingulate. And in parallel, we also monitor non-invasively their sleep state with EEG and EOG and EMG. Okay, so the first thing with these patients, we didn't have the fancy animations movies yet. So they had a learning session in the evening that they had to learn associations between famous people like Marilyn Monroe and animals, let's say like a cat, or Donald Trump and a parrot. Then they had an immediate recall test. Have you seen this person before, yes or no? There were some lures, like Jack Nicholson, that they haven't seen before. And if yes, tell us what was their animal, what was their pet. Then they went to sleep overnight, either one night undisturbed sleep. By the way, is there heating here? Could it be that there's heating? Because I see, as a sleep expert, I see many struggling here. And I see that this is on 26 degrees. So maybe, they went to sleep, one night undisturbed sleep, and on a different night of the week sleep with a closed loop stimulation intervention that I'll describe in a minute. And then they wake up and the order was counterbalanced. Some had natural sleep, then sleep with intervention, or the other way around. And in the morning, we test their memory again. So what is this intervention that we did? For around 90 minutes in the first part of sleep, during deep non-REM slow wave sleep around midnight or 1 a.m., we made sure that there's deep non-REM sleep. We had a neurologist in the room. We were able to identify slow wave, sleep spindles, all the markers of the sleep. And then we delivered intracranial electrical stimulation pulses in a way that was precisely timed with the ongoing field potentials around the hippocampus. So we monitor the field potentials around the hippocampus in real time. When they have a negative deflection, this is when neurons fire more, and we have all the ripples and the replay. And then so they have these slow wave up and down states. And whenever the neurons in the fields indicate that the system is going into an active state, we use that timing information to deliver very brief, high frequency, it's almost like a single pulse, 50 milliseconds in the frontal lobe white matter. So we stimulate away from the hippocampus. We just use the hippocampus signals for timing. And this shows you examples of the location of the probes for monitoring and for stimulation, and technical proof that we were able to close the loop and deliver the stimulations in those times that we wanted to. So what does this do? What is this? I'll just say it's very different than, let's say, DBS for Parkinson's. That's typically continuous high-frequency stimulation, right? Here we only deliver a brief pulse once every 4 seconds. It's very intermittent, but the timing is quite specific. First of all, in terms of memory, we could compare for each subject their memory performance in the night with a stimulation to the night without stimulation. And you can see that in each and every patient, the red bars, the patients who received stimulation that was perfectly timed, not perfect, but synchronized in closed loop manner to hippocampal activity, we saw somewhat improved memory. It's not a massive effect, about on average 10% better, but it was quite consistent across patients. And the interesting thing is, in a control separate group of patients, we delivered exactly the same number of pulses, the same stimulation pulses, but now their timing was completely random, not related to the hippocampal activity. And those are the gray bars where the stimulation actually was, the memory was worse if we did the stimulation in a manner that was not timed. What did this do to electrophysiology? So it did a few things. The first is it enhanced sleep spindles. These are these 1 second long bouts, oscillatory activity between 10 and 15 hertz that we know during sleep has a role in memory and plasticity. And we saw that this stimulation increased the number of sleep spindles, and actually in a quite widespread manner. Let me just maybe see if I have a pointer here. Right, so we see quite a widespread manner. The other thing we looked at is spiking activity of individual neurons, and to what extent the up and down of neurons in distant regions from the hippocampus, let's say in the frontal lobe, are they synchronized to what's happening in the hippocampus? So here's an example where you can see the, well, I'm just gonna, כולם ישראלים, so I'm just gonna like use my Mediterranean hand waving things. You can see that these are the active periods in the hippocampus, and you can see that before stimulation, a frontal neuron may or may not fire in sync with these hippocampal active periods. But in the minutes after stimulation, this is of course a best representative example, one example, but it helps illustrate the point that the neurons had a tendency after stimulation to spike in synchrony with the hippocampal slow wave fluctuations. And when we quantified this, we saw that this was on average true, especially for neurons that were distant from the hippocampus. Before stimulation, they did not necessarily go up and down with the hippocampus. Sometimes yes, sometimes no. But after the stimulation, these two very distant brain regions had a tendency to oscillate in sync. Finally, we looked at the co-occurrence of sleep oscillation. There are many influential theories that say that the precise coupling between slow waves, spindles, and then hippocampal ripples, that they have a phase amplitude coupling relation, that one happens preferentially at a certain phase of the other, and this is part of the mechanism that helps consolidate memories. And we did find evidence for that when we looked at pairs of electrodes in the medial temporal lobe and in the medial prefrontal cortex, which is a primary projection zone of the hippocampus. We found increased tendency for this oscillation co-occurrence after stimulation. And maybe the most important result is the scatter plot, because for behavioral data, we only had 10 to 20 patients. It's not a lot. But it's the best we could do, and it took us 7 years, but it's not a lot. On the other hand, for the sleep electrophysiology, we have hours and hundreds of thousands of events. And what we found is that the more the stimulation benefited memory on the y-axis here, the more it had the effect on electrophysiology on the x-axis. So I think it's quite an important result. And we believe it may be a first demonstration of a causal role for hippocampal cortical synchronization in causally improving memory consolidation during sleep. Something that was suspected from a long line of animal research, and we were able to take it one step further in humans. One major confound should be addressed. Of course, these are epilepsy patients with severe epilepsy. Could it be that what we did is not so much improving memory, but maybe suppress some pathological activity that these patients have that would normally disrupt memory? Or in other words, would it also improve memory if this conceptually was done in my brain tonight, hopefully without epilepsy? So to tackle this, Rotem Falach in the lab focused on interictal spikes. These are abnormal electrical activities that occur in epilepsy between seizures. And it's known from previous work, for example, from the Buzaki lab, that when spikes occur in the hippocampus, they drive activity in the cortex, so they drive abnormal synchronization between hippocampus and cortex. Could it be that our stimulation simply reduced these spikes? So on one hand, we found that it did reduce this pathological activity, an issue that is interesting for clinical applications. At the same time, we found that it reduced this pathological activity to a similar extent whether the stimulation was timed in a closed-loop manner or whether it was not timed. And as you may remember, for the memory, it was completely opposite. One mode of stimulation helped memory, and the other disrupted memory. So we don't think this can be sufficient to explain the improvements in memory. So to summarize this part, synchronization between hippocampus and cortex seems key for memory consolidation, and this process can be amplified by closed-loop intracranial stimulation. Another domain that we have become very interested in is disruptions in both sleep and memory in older age and neurodegeneration, right? In conditions such as Alzheimer's, we have disruptions of sleep, and we have disruptions in memory. Could these things be related? And what goes on? And we work with many people at the Tel Aviv Medical Center at Ichilov, neurologists, neurosurgeons, to tackle this. We work especially with mild cognitive impairment, MCI, also Alzheimer patients and Parkinson patients. They come in for overnight sleep studies. And before we became interested in this, it has been established that the slow waves during sleep are different in their amplitude. This is just one example for how slow waves look in a 70-year-old healthy individual or in an Alzheimer patient. You can see with your naked eye that it looks very differently. The amplitude is reduced. And indeed, this goes hand in hand with increased pathology, as can be seen with PET imaging in this case. Our question was, okay, can we also identify these changes in sleep earlier in the prodromal stages of the disease in mild cognitive impairment? Because, you know, when a person has advanced Alzheimer's, the massive degeneration, there's less gray matter, so everything is different, so the slow waves are different, so it's not that surprising. Could it also be happen earlier in the disease? And we found actually that specific features of the sleep oscillations, in this case, the negative, the amplitude of the negative half peak, if one identifies this carefully after cleaning the data, then first of all, it's the amplitude is larger in control subjects, reduced in mild cognitive impairment, and further reduced in Alzheimer patients. But even more interestingly, this shows this can be pushed to some extent to the individual level. So if we track the amplitude of the slow waves for each individual on the x-axis, they actually correlate quite well with the cognitive score here with a MoCA questionnaire, but also with other tests. So sort of give me your sleep wave amplitude when you're 70, and I can infer something about the cognitive status. And now we're pushing this not only to the current situation, but also to look at prognosis. Will this say something about whether some individuals deteriorate more rapidly than others? Another thing we're very interested in, and this is Ina Slutsky is to blame, is that in animal models of Alzheimer, there have been reports of abnormal hyperexcitability in the hippocampus that's accompanied by interictal activity, epileptic-like activity in the hippocampi, right? Hippocampi of Alzheimer mice. So here's one example from the group of Ina Slutsky where when mice are awake, when healthy mice are awake, their EEG is very active, high frequency. When they're anesthetized, they show this burst suppression activity. When you look at Alzheimer mice, when they are awake, it seems very different to healthy mice. But all of a sudden, during light anesthesia in this case, but also during natural sleep, they start showing these spikes that reflect hypersynchronous hippocampal activity. And actually, it's a very dramatic effect. Could this also happen in early patients, in real people that have Alzheimer? There have been some reports that yes. This, for example, is a paper from Harvard, published in Nature Medicine, where they somehow had access to two or three Alzheimer patients who had electrodes at the base of the skull that were able to record activity close to the medial temporal lobe. It's not really in the brain. It's close to the base of the skull. And what they reported, I hope it can be seen, is that around the left hippocampus, you can see these spikes, but they are quite invisible in other more distant locations or non-invasive. So this spurred an entire field of curiosity and research. Could it be that in real Alzheimer patients, below the surface, deep in the brain, there is lurking epileptic-like activity? If this is true, it has a lot of implications. Imagine, we have, as a biomedical community, we have a lot of tools to try and battle epilepsy. We have devices, we have drugs. Could, these patients, maybe we maybe they could benefit from these anti-epileptic treatments, and maybe this would help their memory. So this has quite a lot of implications. But can these hippocampal spikes be detected non-invasively? It's a controversy. Some groups, like Keith Vossel and others, reported that yes, 20-30% of Alzheimer patients, we can detect this even with EEG and MEG. But other groups said no. It happens in, let's say, 5-10% of individuals, and it's similar in healthy 70-year-olds or in Alzheimer patients. It's no different. And we were frustrated when we came into this question that the entire work is done by visual inspection of the EEG, neurologist style. You can only see so much with your eyes. So we thought, could we do better? And we went on a long journey. It's been 5 years now, led by the amazing student Rotem Falach, and in collaboration with Vlad Zelezniakov, the neurologist at Ichilov, and Amir Globelson, who you may know from his ELSC training days, machine learning expert. And what we did is we used the fact that we have this golden simultaneous data recorded in the hippocampus of epilepsy patients, and at the same time non-invasively. We thought, could we train machines to pick out features with the non-invasive signals that could identify moments when something happens deep in the brain, right? Because we have the ground truth. And let me start by a teaser at the end of the process. The machine said, this is a 10-second trace of EEG outside the brain, non-invasive. Our machine says, yes, somewhere here there's an interictal spike deep in the brain. Can you spot it? Can you try to guess within yourself? I don't know. When would you say this event happened? It's not easy, first of all. So our machine says it happens right here. Not sure whether this would be the moment you choose. Maybe yes, maybe no. The thing is, we have the hippocampal field potential to say, okay, in this case, the machine was correct. And of course, it's not magic. There are other events like this one that have lower amplitude, probably a more focal event that doesn't leave any trace non-invasive. And after years of work, here's what we did. We took data simultaneously from the hippocampus, from, let's say, standard scalp locations up here, but also from the upper cheekbones on the zygomatic bones. And to our surprise, it was mostly these sensors, these electrodes here, that carried information about medial temporal lobe activity. So the way we worked, we wanted to build a large cohort because these patients are very rare. To train our machines in the most powerful way, we teamed up with a center in Bonn and also in Milan. And we got access to over 50 overnight recordings where we have more than millions of these interictal spikes. We trained the machines. Then on the second stage, we brought epilepsy patients only to non-invasive EEG studies. But note that our nets also have sensors in these regions here that are very relevant. And then finally, we'd like to take this machinery and go back to early Alzheimer and address our question. Is it true that in real Alzheimer patients we can find this interictal activity? We used a XGBoost algorithm of machine learning, and we can achieve now a precision of over 80% and a sensitivity of about 17%. So we can detect 17% of the events. If our machine says something happened now, it is accurate in over 80% of cases for new, untouched individuals. So these are other individuals where we perform the test. And now we can bring epilepsy patients to the lab that have mesial temporal lobe epilepsy. These are patients that the neurologists say it's impossible to know what's happening in their epileptic activity. You need to implant electrodes in the brain to see, right? It's those type of patients where it's difficult / impossible to see things on the outside. We just give the data to our algorithm, compare it to age-match healthy controls. We focus on these sensors here. And now we can reach a situation in one cohort on the left and in a completely untouched validation cohort where we lock all the weights of the machine and all that. And our machine can say in quite a reliable manner who is a mesial temporal lobe epilepsy patient and who's a control. Again, in data that the neurologist can barely identify anything visual, right? So what I'm trying to say is the EEG has much more information than what we can easily identify with eyes, and especially if we have solid ground truth to train it on, then we can do so much more with the EEG. Now for the first time, this is 3 weeks ago, very fresh out of the oven data. We've finally used the same machinery that we've developed and validated in epilepsy. We took it to elderly, mild cognitive impairment individuals, and you can see that on average, the occurrence of these events in MCI patients is higher than in the controls. And interestingly, when individuals advance to it's not the same people, but a different group where it's more advanced Alzheimer, then we don't necessarily see this signs of deep interictal activity, and we find diffuse initial signs of some correlation with the cognition. So we believe this would be significant. We have a way now of identifying in some early Alzheimer patient this epileptic-like activity deep in the brain. And we're now excited to check whether this can, for example, define a new subset. Maybe we call all of these Alzheimer patients, but maybe some of them do have epileptic activity and some do not. Maybe some can benefit from some drugs, different than other individuals, and so on. And by the way, epileptic activity is not only happening in epilepsy patients or in Alzheimer. It can also happen in other neurological conditions, for example, children with autism or individuals after traumatic brain injury. So we think that this tool can have wide applications for detecting epileptic activity in all sorts of neurological conditions, and we're excited to look at that now. So here, I need to do a sharp transition to the pupil. Yes, Aviv.
How common is to see epileptic-like activity in control?
Fair enough. So, it's a good question. We're not sure. With the eye, with visual inspection, people report in elderly, it also depends on age. In elderly people report that in 5 or 10% of individuals, healthy individuals, some activity along this line can be identified. Now for the first time we have this tool that's probably a bit more sensitive and looks deeper in the brain, and we have this result. Let's say these healthy individuals that have a relatively high detection rate of our machine. The thing is, we don't have now the ground truth, right? We don't have electrodes in the brain to say, is this 100% accurate? So I guess it's an open question, because we don't really have good tools to measure the electrical activity deep in the medial temporal lobe non-invasively. This is getting a bit closer than what we have had so far. So it lacks the ground truth. I'm not sure.
It exists, right? Suggested it exists in everyone.
Yes. Well, at least our machine has some rate of detection per minute in everyone. But then, it could be false positive. So I would, personally, to be conservative, I would put the emphasis, let's say if we could draw an imaginary threshold that is very, very rarely exceeded in healthy individuals, and some MCI patients exceed that threshold, then I think that thing is probably meaningful. But whether most of the cloud here is meaningful, I don't know. Yes?
Is the variability due to differences between people or in the same person? You see that sometimes the algorithm will detect more spikes and sometimes less. Let's say you divided the data in half. So I'm wondering whether it's the ability to detect that differs.
So we have now 1 night per individual with these data, with these cheek data, right? It's not standard EEG. And we do see variability across the night. But also we know that epileptic activity varies across the night as a function of circadian time, as a function of depth of sleep, and other factors. So there is variability within individual, and there is also variability between individuals. I'm not sure it's a satisfactory.
Maybe it's the additional differences looking at the variability.
That's a good idea. That's a good idea. We haven't done that yet. We just put 1 score per night per person so far. It's a very good idea. All right, I think the ultimate test would be if those individuals with MCI that do show very high levels of detected events during the night, if their memory progresses differently. If, let's say, we look carefully now when we find structural abnormalities in their MRIs, if they respond to some drug differently than other individuals, these building blocks may with time increase our confidence in this result, which is still preliminary. This is how I think about it. All right. So at first I had like 15 minutes on the pupil and the locus ceruleus and noradrenaline and mice and fiber photometry and optogenetics. A very wise friend told me, oh, ditch all that, so I did. And I will just say this. During sleep, things are not static. Even during non-REM sleep, there are changes in the depth of sleep. And recently, there came out a study in Nature that separated moments of non-REM sleep depending on whether the pupil in mice was dilated or whether the pupil was constricted. So there are changes in arousal and in noradrenaline signaling during non-REM sleep, and it was shown that actually memory replay is very different in these moments or in those moments, especially these moments where the pupil was constricted and sleep was deeper with less noradrenaline was particularly important for replay and memory consolidation. So the pupil and its changes during sleep are very important. And as a lab who's interested in both sleep and noradrenaline, we always were fantasizing about could we record the pupil during sleep through closed eyes. And I'm glad to update that we have some achievements along this line. First of all, this was a project that was my baby pet personal project. I used to come to the labs on weekends, have mice, infrared fibers trying to illuminate through their ears, through their skull, through the... I did a bunch of, is the IACUC committee here, I tried a bunch of stuff until we teamed up with experts in biomedical engineering, light tissue interaction. And through our dialogues, this is work led by Omer Ben Barak and Barak Haddad with Michal Tepper and Professor Israel Ganot at bioengineering. And we used a specific wavelength frequency band in the infrared range that's called shortwave infrared, or SWIR. SWIR. And this wavelength, it's known, has a better ability than other wavelengths to penetrate into biological tissue. For example, as you can see here when imaging blood vessels through a mouse skull. So with this, we wanted to see could we use that to track pupil through closed eyes. And we wanted to start with the most robust phenomena. The most robust phenomena you can have with a pupil is its response to flashes of light. You've probably seen this, like in all the House and all these medical series that physicians often use a flashlight. When you shine light, the pupil immediately constricts. Let's say if I have like an iPhone flash, pupil constricts and then it grows back to it dilates back. And this has a very, very stereotypical time course that you can expect. So we thought, okay, let's zoom in on that. And we had experiments where participants had one eye that was, I don't have the setup here, one eye that was open, and we had standard eye tracking, gold standard eye tracking. We knew the pupil at that time was 4.15 millimeters for sure. And then the other eye we had closed. We had this infrared device, and we tried to develop algorithms to get the pupil size. Here you can see, and this is really representative data, I mean after years of work, where you can see here in blue is the open eye. So every time we flash a light, these trials, you can see the pupil reduces in size and then it dilates again. And in orange, you can see the situation of our SWIR imaging with closed eyes. And this is, by the way, a control region on the forehead, because maybe some illumination changes and the level of brightness or something could affect the result. When you look at the result averaged over 40 participants, In blue is the pupillary light reflex in the open eye, and in orange is the PLR, pupillary light reflex, in the closed eye. It's not the same units, it's not the same amplitude. It's about order of magnitude weaker, right? So let's say a 10% change in the open eye corresponds to a 1% change in the signals in the closed eye. But the time course is very, very similar, and we can track this in individual subjects. Another thing we did is we played around with deep learning algorithms, in this case a unit model that transforms one image to another image. So you train it on pairs of images, and it calibrates the weights to optimize the transformation between one image and the other. So what we did is we trained it on pairs of images where one was an open eye and the other one was a closed eye. And here you can see this is the real open eye, and anyone working with deep lab cuts can see the circles that denote the pupil. Here is the estimation of the model. It's based on closed eye data. What does this think that would happen were the eye open? And here's a control region. So let me show you how this looks like after training our models. Oh, I need to get rid of this. Not sure how I do this. Escape. Thank you. Bright minds. Okay, so soon the flash of light will come. This is like the experimental trial. Look at the pupil. You can look either on top. Right now, the light comes, the pupil constricts. This is reality, and this is our closed eye data. You can see that on a single trial level, it can track the pupil. It can identify that the pupil constricts and goes back again through closed eyes. Here's another trial. And this does not happen when we train the model on a control region. So with this, not only can it track, by the way, pupil size, it can also track gaze direction, where the eyes are pointing to through closed eyes, to make both horizontal movements, vertical movements, in very controlled experiments where we ask the people to gaze at different points on the screen. And we can now achieve a level of error of plus minus 10 degrees. Which is not perfect, but imagine this is 90, 45, 22, like this. We can say within this error where the eye are looking at during when the eyes are closed. This is a touchless device. It doesn't touch the subject. It can operate continuously for hours. There's no heating. And we're it's a very methodological advance, engineering advance. It's a tool, but a tool we're super excited about because we think that it opens new horizons, first for research. You just saw that Nature paper in mice, that everything is different in sleep when the pupil is small or is big. Could we do those type of experiments in people? And also clinically, we think that this could be useful in anesthesia, at the intensive care unit, for sleep medicine. We don't know how far we can get with this, right? With further improvements in the SNR, could it be that pupil dilations during sleep can give us a window into a host, an array of arousal events during sleep? Imagine that you could detect a seizure like this. Maybe you can detect a heart attack during sleep, hypoglycemia during sleep. Maybe you can detect intraoperative awareness, or a person is unnecessarily feeling pain. All these types of things we're tantalized with the potential of this, and time will tell how far we can go with this. So with these sort of boring technical developments, but I am biased and I'm excited by them, I'll wrap it up for today. I'd like to thank the team at Tel Aviv University, colleagues in Israel and abroad, the team at the Medical Center, Ichilov, the funding sources, and thank you very much for your attention. Lots of tools, nothing new on how the brain works. Yes, Leon?
Okay, it's technical, so I ask a technical question. How sure are you that the zygomatic EEG is not zygomatic EMG? You're not recording muscles in the face which are correlated with the spikes in the hippocampus rather than directly with the brain.
Fair enough. First of all, we see first of all, it's not something that we notice in other electrodes that we do have on the chin that also record. So if there was a global facial twitch or movement, I would imagine that we would have seen something similar in other locations. If it's a specific, if you're saying, how about a specific muscle twitch over here, I'm not sure we can rule that out. But I would say that if, let's take this to the extreme. Let's say every time there's a hippocampal interictal spike, it so happens that something twitches only here, and we can measure it.
Great biomarker.
Then I think for most of the purposes that we are interested in, it's a great thing. I don't think we can rule that out. Yes?
Hello.
Hello?
Question. Many times, actually, the U-shape you mentioned about MCI and AD
Yes.
is quite common phenomena, if I'm not mistaken, because we see kind of, we call it adaptation or compensation effect in MCI, and then we see that they are not able to compensate. I remember seeing it in many EEG results about MCIN. So you probably have some interpretation. Do you find it as a compensation effect, or you can interpret it?
So I can only speculate. I mean, I don't think our data or our study, to be honest, are the strongest or most suitable data to address this question. I think that either, let's say in animal models where you can really track these animals longitudinally over time, we know, for example, because Ina Slutsky is my neighbor, so I know their data and their work, they also see similar things, that this is, as you say, particularly indicative of earlier stages of the disease, this hypersynchronous aberrant activity. I'm not sure about the cellular or the mechanisms that drive this compensatory activity, or how it collapses later on. I can just say it's in agreement with other data, both from patients and from animal models. You are right. But what drives this thing at that specific phase of the disease progression, I don't know. I'm not sure. Yes.
Two questions about the memory
Yes.
study. One is whether you looked at pupil size. I know
Yes.
it's tricky. So
We did. It's online in the preprint. The pupil size, we found orthogonal effect that had to do with familiarity. If you were familiar with a movie, there were effects in the pupil, but these were not observed specifically around those events. These gaze effects are specific to the few seconds before the event. So we found pupil effects that go with familiarity, while these specific gaze effects go with memory of the event. I should also say, I didn't mention it, skim through it, that there's also some of these gaze effects when people report they do not remember. It's not a perfect correlation with memory, which is on one hand interesting. People say, have you seen this movie before? No. Are you sure? Yeah. Haven't seen this movie before. But their eyes, the way they look at the movie, tell us that they remember the meerkat is about to jump behind that rock. And on one hand, it's very interesting to measure unconscious memory. And on one hand, it complicates our ability to infer from this measure about the report. You see what I'm saying? Yes.
And do you see information in the eye gaze in the first presentation that will predict whether they remember?
Not so much, but this I think is mainly because we tailor-made the animations in a way that there's almost zero chance you will look at that rock the first time you see the movie. It's very different from real-life movies. We've also done real-life movies. It's all in the preprint. But these animations are sort of maximizing the contrast between the first and second viewing, meaning they are compiled in a way that will reduce chances to nearly zero that you will look there in the first time, and maximize the chance that you will look there the second time when you remember. So
it's
it's
very
deliberate.
brief. After the meerkat jumps, it ends quite quickly.
Yes.
So you don't have.
Yes. But we can discuss more. Yes.
Do you intend to apply this kind of memory studies to dreaming? Kind of wake up participants and see if they report anything?
Do you mean for the closed eye imaging or for the movies, or what do you have in mind?
I was thinking about the movies, because you have this like intervention during the night where you simulate the brain, and then I was wondering if you would consider waking up the patients after the simulation.
All right, so you're connecting a few dots that in our research have been so far unconnected so far. The project with epilepsy patients where we did the intervention did not yet have the movies.
Oh.
And you are also suggesting to wake them up and ask them about the dreams. It's something we always wanted to do for now 20 years. It's a very, very difficult setup. I mean, these patients are hospitalized and yes, they volunteer for research, but most of them are not super happy to be woken up during the night to interrogate about their dreams. We always wanted or were hoping as scientists to go there, but we didn't do it. We didn't do it.
But for the movies, you can wake up patients, subjects, let's say during slow wave sleep or after
Healthy
school
people.
Or, yeah.
Yes.
Do you intend to do it?
Whether they dream about these movies, is that the...
Yeah, or if they report anything, like more dreaming in general.
We weren't planning on going there. It touches on an entire domain of to what extent our dreams are related to content that we experience shortly before sleep or during when we were awake. You'll be surprised maybe, but the vast majority of studies that looked at this find very little relation, at least explicitly, if you're not Freudian, between the visual content of a dream and the content that you experienced just before when you were awake. Apart from some Tetris that people, did this for like hours and they really close their eyes and they see these things falling, it's quite rare. Yes?
When people see many of those movies, do they change their patterns, start wishing for something to happen somewhere?
Many different movies or?
Different movies, they expect something to happen.
So it's a good question because it also touches one of the reviewers, maybe that's you, asked about statistical learning and whether, let's say maybe when you see the 20th movie, you already get it. I shouldn't look at that object, the giraffe they're showing me. Something else is going to happen. I know these guys. And we start, gazing around and things like that. We didn't find any evidence for that. Actually, a reviewer asked us, and we just did this analysis this month. So, no evidence for that. Yes?
Are there cases where the subject tells you very confidently, yeah, I remember this, but there's nothing in the case at all?
Good question. We were always interested in the mirror image of that, and I don't think we really looked at that. I would imagine that, yes, you could be super familiar with a movie. Okay, I know the meerkat is going to jump, but so what? It's a boring experiment. I'm going to look at the leaves. So I would logically imagine these trials exist, but we never analyzed them in detail. If any of you have ideas for how these new tools could be brought to their full potential in experiments that we didn't think about, I'd love to hear that after the seminar.
So, great. Thank you very much.
No. הצורה שאני מדבר את המילה.
זה מדהים, זה פשוט, אני יכול כל כך, אתה יודע, בניסויים זה מאוד מאוד אנחנו האמת ניתחנו חוץ מזה, יש לי עוד הצעה אחת שאולי תספרן אותך, קראתי קצת בעימות המוטיב, יצא אותי מערכת תנועה של שוט, בעצם מסיקה מזכירות קוגניטיבית בדיוק מטעם סיריס. יש לי וידאו,
אמרתי, היי, go sell it to Audi.
כן, לא, זה ממש מסכימים, כל פעם כאלה, יש לי עד היום, אבל לא משנה, בכל אופן הם כלום אופציה. אבל...
כל כך שמח להגיד, זה משהו של המון התחומים.
כן, תודה מקרב לב. אמרתי לפני שאני
אז נהייתי עם
זה היה עונג גדול.
תודה רבה. מה היית השם? אנה, מעולה. אז אם תוכלי לשלוח לי אימייל, אפילו אימייל שהוא יכול לשחרר.
מה זה? הוא הוועדה שלי.
אני לא עובד בשביל לך על מוביז. מוביז על ממורי, מוביז, ואני אחבר אותך עם פלאביו שהוא הפוסט-דוקטורנט, הוא ישמח מאוד להדריך אתכם, to set it up, הוא יבקר פה הכל. אנחנו לא נהיה מאושרים מאיתנו שתשתמשו בזה, we'll help you set it up.
בשאלה אחרת, בנושא אחר, לגבי הגייסיסטים, אמרת שזה שיטת המדינה שלו.
כרגע, כן.
אז רציתי לשאול איך אתם עושים קליברציה?
לא, אז הניסויים של האלה הם עם עין אחת פתוחה ועין סגורה. יש טארגט שמופיע על המסך, והבן אדם עוקב אחרי זה, ואנחנו משווים, זאת אומרת...
כלומר, הקליברציה היא בעצם על אדם ש...
כן, זה שואלת שאלות נכונות. כרגע עשינו עדיין דברים שהם יחסית יותר קלים. עד עכשיו כל הניסויים שלנו, כמעט, לא סיפרתי אחרת, זה שעין אחת פתוחה ועין אחת סגורה, ואנחנו מניחים שגם העיניים מסתכלות לאותו מקום, בבן אדם שאנחנו מבקשים ממנו, וגם שהאישונים הם לגמרי סימטריים, שזה הנחה סבירה בבן אדם בריא, במצב נורמלי, ואנחנו בעצם לוקחים את הדאטה של העין הפתוחה כ-ground truth בשביל העין הסגורה. זה היה הפרוט שלנו. צודקת שבשביל להתקדם עם זה הלאה, למצב ששתי עיניים סגורות, ואתה עושה איזה estimation, עכשיו אני חושב שהבן אדם, העיניים שלו מצביעות לשם, אז אין לך בעצם את ה-ground truth. אתה צריך לעשות איזשהו leap of faith ממה שעשית במצבים של פתוח סגור, למצב שסגור.
גם בממצאים של
יש פה איזו קפיצה שצריך לעשות.
אצל חגי גם הוא מראה שבחלק מהשאלה...
העיניים לאו
דווקא.
אבל כרגע אנחנו לעשות את זה...
כן, כשהרים לזה.
יש אנשים
לעשות את זה כל עין בנפרד, כן?
יש אנשים שיש להם לחץ תוך עיני ושמים להם מין נתזים כאלה, שזה מין צינורות מתכת. יכול להיות שיכול לעקוב אחריהם עם שדה מגנטי, ואז לעקוב אחרי תנועות עיניים שעיניים.
אנחנו
זה גם לשם.
כן, אנחנו רצינו לפתח משהו שיוכל לעבוד בכל בן אדם.
לא, בשביל לקבל כמה פלוס לשתי עיניים.
אבל שיש, כן, מלא קופים מושתלים.
לא, בקופים ברור שיש.
כן, אבל זה דרך אגב, זה
זה
לא ביגוד אינף, למשל, אם לחגי ברגמן פה יש קוילס שנותנים ב-100%.
יש לו מלא
מה זה
דאטה
ב-
קוילס, כן.
ואנחנו נשים במקביל, בנוסף, את ה-device שלנו פה. אני חושב שאם זה עובד טוב בקוף,
אני אגב לא...
סיבה להניח שהעפעף בבן אדם הוא מאוד שונה.
בקופים העיניים שחורות, אין את הרמה, לא יודעת אם זה משהו.
אוקיי, אז אולי זה כן משנה. אני יודע כל כך מעט על הקופים, אולי זה כן משנה. erase all that, scratch all that.
עכשיו איזה קוף?
fair enough, זה הכל שאלות נכונות. יש גם אנשים שיש להם מה שנקרא, שהעפעפיים שלהם לא לגמרי נסגרות עד הסוף בשינה, שהם סוג של ישנים עם עיניים פתוחות, חוץ מזה הם בריאים, זה בדרך כלל דבר לא טוב כי זה מאוד מייבש את העין, קרנית, וצריך לטפטף טיפות, ולפעמים עושים להם איזה כירורגיה סביב זה, אז גם זאת יכולה להיות נעישה. כאילו לזהות אנשים שגם ככה העיניים שלהם מאיזושהי סיבה כזאת או אחרת פתוחות, או יש לך הצצה פנימה. שוב, נשים את הדברים בפרופורציה, כרגע יש לנו ניסויים, אנשים, ערים, מתנדבים, בצ'ינרס, במעבדה. עכשיו אנחנו לוקחים את כל הדבר הזה לעולם של bedside, בבית חולים.
לא מזמן היה מאמר ממון רך. לדבר עם יואב אדם, שכחתי את השם של זה, שזה חומר כזה.
כן, הדוריטוס.
כן, בדיוק.
זה, אני יודע.
זה יכול להפוך לך את הפעמים לשקופות לצורך העניין, בשביל לקוחות.
כן.
אומייגאד, מה זה החומר?
זה סתם, זה מין חומר אדום כזה צבען.
שיש אותו בכל מיני ג'אנק פוד?
כן, ושאפשר, שיכולים לראות דרך, כאילו הופך את הטישו לשקוף.
בגוגל, brain, transparent, דוריטוס, enter, אתה תמצאי.
כן, זה הופך את הכל שקוף. נכון, אתה צריך ללכת
קודם כל, באמת, היה לי 8 סליידים, ממצאים חדשים על הלוקוס-אוריוס ונורדרנלין בעכברים עם אופטוגנטיקה ופיילופוטומטרי וכל הנפלאות של הזה, ונפגשתי בבוקר עם חגי והוא אמר לי, אתה מתנהג כמו ילד שבאת להרשים בפלופטוק, אני קורא את האבסטרקט, את סטומאץ' בוא ניתן לך הצעה של חבר.
זה עצה של חגי, לא, אבל סבבה.
תדבר על פחות דברים, כי אחרת... כן, אני עכשיו... קודם כל יש לי כמה דקות עכשיו.
כן. לא, הבנתי את זה, לא, פשוט...
למה אני אומר? אני עכשיו לקחתי, אם נגיד את זה החלטה טובה, לא, זה תפקיד חדש באיכילוב, אני רק איזה יום וחצי בשבוע בקמפוס.
באמת?
אין להניח שאתה בקמפוס, אני שם.
מה...
באיכילוב, בעצם אני יורש את תלמה הנדלר, שאני יודע שם את
למשל התוכנית של זה.
כן, היה מטריבאוט, מה שהציעו לימון, וכן ולא, וכן ולא, בסוף לא הסתדר, אז אמרו, יאו, הוא אומר לך בסדר. יובל! כן, כן. ובנוסף, לא במקום, בנוסף לכל מה שיש לי בקמפוס. בקמפוס יש לי...
צריך ריבל 1 וריבל 2.
זה לי ספירה כרגע שלא... מה?
ויצא לך קפה?
ואני מספר לעצמי, שעוד שנה, שנתיים, 3, אני אהיה קורסים אלפטים, ואני אענה מכל. אבל כרגע זה קשור כלומר, יש לי כאילו, גנרלתי, ואחרים,